All right, here's our last mini lecture from chapter 9, and it's the last piece, and it's on fermentation, and we'll see that we're used to thinking about fermentation with regard to things like brewing beer, but it's the same term and the same principle that happens in our body when we run out of oxygen and we can't do oxidative phosphorylation. When we can't do oxidative phosphorylation, all these chemical reactions build up, and fermentation is a process that happens in the cytoplasm. And it is how the cell is able to continue glycolysis and the production of some ATP in the absence of oxygen. So compare the presence of oxygen, highly efficient production of ATP. In the absence of oxygen, only a little bit of ATP gets made, and the NADH that gets made during glycolysis is actually thrown away. And we'll talk about why it's thrown away. And so fermentation is a very inefficient process, but it continues because, for example, when we're running and our muscles run out of oxygen, we want to keep running because maybe we're running away from a tiger, like that, you know? So it's kind of a life-saving shunt, and it's included in a chapter line a little bit to show you how there can be shunts or alternative pathways in metabolism, because there's a lot of that happens in our body, switches. And so this is just really a metabolic switch. And in this case, it just is a switch that occurs automatically because of a great big traffic jam. And so you'll see that this pyruvate is going to build up because it's not carried into the mitochondria. It stays in the cytoplasm, and everything about fermentation happens in the cytoplasm because the pathway into the mitochondria is blocked because of a big traffic jam. And so here it's just showing that in the presence of oxygen, cellular respiration takes place in the mitochondria. In the absence, pyruvate takes an alternative pathway, and through a couple more chemical reactions is broken down to either ethanol with the release of some CO2. Ethanol is a 2-carbon sugar, or an ethanol fermentation occurs in yeast, in bottled beer, like that, or wine, so alcohol fermentation, or lactic acid that builds up in our muscles during exercise, like that. And so it's just a switch path, a branch point in metabolic pathway. And here's what's going on. This is all happening in the cytoplasm, or cytosol. We still have the normal glycolysis occurring with the normal steps, and we're producing ATP and NADH. And the key to fermentation is that when this occurs, remember that there's a fixed amount of NADH and NAD+. So this is the key concept right here. There's a fixed amount of that in the cell, and it's being recycled. And normally it gets recycled by having this NADH go to oxidative phosphorylation, and oxygen is used. But what if oxygen is limiting and you can't do that? What happens is that fermentation provides a shunt to recycle NADH in order to keep glycolysis going. It's kind of amazing, but if glycolysis was continuing without this recycling, we would run out of NAD+, and glycolysis would stop because one of the essential substrates in a chemical reaction in glycolysis would be missing. And so this lower part of this fermentation pathway provides a way to recycle NADH. And in doing so, this energy is just lost. But it allows for glycolysis to continue. One of the products of fermentation could be ethanol. Here's the structure of ethanol in alcohol fermentation. And I think the other pathway is the production of lactic acid in our muscles as a different form of fermentation. Look how similar the two pathways are. In both cases, the goal is to keep glycolysis going in order to keep producing a little bit of ATP so the cell can stay alive and the organism maybe can survive. And in doing so, just throw away this energy in recycling the NADH. So to go from a very efficient process, capturing energy, including ATP here in the presence of oxygen, to a very inefficient process because we're just throwing energy away. How is it thrown away? This is really interesting. So this energy gets thrown away in the form of heat. So remember that when we're releasing energy, we can measure it in calories or joules. Calories are heat, joules are work, they're interconvertible. And we can use a fire in the wood stove and we can just throw everything away as heat. Or we can capture some of it as work energy in an engine. And what's happening here is that oxidative phosphorylation creates a very efficient cellular engine. And we can capture that energy and we can capture it without losing much heat. When you start exercising, you start sweating, you're producing a lot of heat. And one of the reasons you are is because oxygen in your muscles becomes low and you start doing this and you lose more and more calories as heat and heat loss, like that. Some of the calories are captured because this lactic acid or ethanol, this lactic acid is eventually converted in the presence of oxygen. So there's energy stored here that's going to be used later. But lots of energy is lost in heat, in lactic acid, in fermentation pathways. And so that's just an example, and the key concept in chapter 9 at the end is the idea of choices in these pathways and regulation of these pathways. And one pathway is regulated just by having the components accumulate in the mitochondria, blocking entry. There are other ways of regulating these pathways, and one of them is shown also here. There's the pathway, there's the citric acid cycle and glycolysis, showing all kinds of different pathways around it, and other metabolic pathways in the cell. And this is the last concept we'll deal with, and that is that some enzymes are regulated, and so the pathway is switched based on the enzymes regulating the chemical reactions. And if we shut off one enzyme, then the material will flow down the other pathway. And this just shows examples of two ways of inhibiting an enzyme. There is something called a competitive inhibitor. Competitive inhibitors bind to the active site of an enzyme and physically block the substrate from binding. So here's a competitive inhibitor blocking access to the substrate, and so it's going to stop that chemical reaction. Alternatively, some inhibitors are called non-competitive inhibitors, bind to another site on the substrate, change the shape of the active site, and reduce the affinity of the enzyme. The effect is the same. The competitive inhibitor and the non-competitive inhibitor shut down the chemical reaction. And so that's just another example shown in Chapter 9, and we need to get more out of Chapter 9 than just what I'm talking about in these mini-lectures, but the mini-lectures provide a good framework for getting you through Chapter 9 and focusing on what's the most important in Chapter 9. And that's it.